Hello!
Hi there.
Welcome to Microbiology Twitter Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology. I've mostly worked on bacteria, but I've also since then worked as a research theory specialist, and currently working as an editor for a science journal.
Every week, we meet to talk about microbiology, and today is our news week, where we do an overview of the papers that we've seen in the news and other sources to try to find an article that we want to cover next week in more detail.
Yeah, next week will be our deep dive week, where we take one of the papers we talk about this week and we take a deep dive, figure by figure, to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
You can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us using the hashtag MicroTWJC hashtag.
All right.
So, yeah, this week we've got a lot to talk about. So last week we did our top 10 of all non-COVID papers, so there's been a little bit of backlog of news stories, but I think the one thing we'll start off is some of the new strains and variants. So,
Yes. Yeah, I feel like we didn't talk about a lot of these different ones. The first
mm,
one is a paper that I guess is close to my home in New York, and it is doing this genetic surveillance. We've talked about the way in which people find these variants, right? Is about sequencing the different the actual viral isolates that come out, and the more sequencing that's done, you can start building out these big phylogenetic trees, right? And in this particular paper, they just did a lot of sequencing in New York City specifically. And from all that sequencing, they were able to add like a separate branch, right? They were able to argue for the distinctiveness of a branch of that family tree of SARS-CoV-2 for this area.
yeah, this has been quite interesting because what they did, they essentially wanted to find out, like, okay, where are there new strains in New York?
Mhm.
And usually what people have been doing is been doing the spike protein dropout. So if you sequence like these new strains, some of them don't, the spike protein doesn't show up. Here, they decided to go, okay, let's actually just sequence the whole genome to see what's happening, to see where these mutations show up. And what they're expecting was to see, oh, some of these old mutations from like places from like the Kent variant and South. They're hoping to see that, but what they found was an entirely new variant,
Yeah,
which
I think
is
that's like there's a narrative of like where do the variants come from, right? Like it's like, oh, it's this global travel. They're like everywhere, right? They're being mixed and matched, right? Like we're mixing the viruses around in the world, and there's some truth to that, right? Like if you go to your local like epidemiology, right? Like I think a lot of localities report something on like the variants that they find in their region, and like sometimes they do full genotyping of those, and then they find, oh, this is the strain that came from X, Y, and Z. But that doesn't, like, for every time that they say that this strain comes from so and so, did they do the whole sequencing, or did they just do like a short little read? I'm not actually sure. I think that that's like something that gets lost in the details sometimes.
mm,
So for them here, for us in New York City, to see this data where they did do whole genome sequencing and say, oh, actually, like the same mutations that you'd find right in some of the other strains, they just like re-emerged in their own way here, right? Like, and I think that that's a great story about evolution as well. Maybe also a warning for people around the world that if you have a high level of viral replication, if there's like, right, if it's beneficial evolutionarily, which is what we're saying when we say the strain is more transmissible, right? Like that's beneficial from the evolution standpoint. Probably you'll see your own version of it pop up, right? It's not going to be really, it won't have the same other random mutations that maybe don't affect the phenotype as much. Accumulate in it, but it could have some of those mutations that do affect the phenotype.
yeah, and that's something that we should all be aware of because not everyone has like great genome sequencing facilities. So there are some areas we have like lots of new strains coming up, but we won't necessarily spot them until they travel to other areas with those facilities.
Mm-hmm.
So sometimes when you hear like things about like Kent variant, South African variant, it might just be not so much the place where it evolved, but the place where it was first discovered.
Mm-hmm.
So and
Exactly.
that's how you think. So um,
Yeah,
Yeah.
so
So
basically, I don't know if this is, I guess it's sort of a bad news story, right? That like, oh, we have our own, but I think it's very instructive from that evolutionary
mm,
standpoint, right? Just helping people to think about like the variants, not just as like coming from a place, right? But possessing a set of nucleic acid right sequence that is beneficial. So that means if it's beneficial, it could appear in other places, right? Especially if your rates are high. Also good. I mean, it's always good to just keep the transmission rates low. It's right, like
mm,
even when as the vaccines are rolling out, like it's not nice to see a lot of viral replication in a country or in a locality.
right. So now we've got another paper which is on SARS-CoV-2 exposure in escaped mink
Yeah,
in Utah, USA.
this is great. I think we might have covered a paper or like a preprint a couple weeks back where they actually captured wildlife and infected them with
Mm,
SARS-CoV-2. But in this one, they're capturing them and they're doing surveillance, right? Yeah.
yeah,
They want to know like I think there was an incident where, or there are mink farms, right, that have SARS-CoV-2, and we heard this news story a while back of like culling of mink farms. Um,
mm,
and what they're saying is that mink can also escape these farms, right, and enter the wilderness. And in doing so, they may transmit any diseases that they hold to the wild population. And so there's some surveillance that they did in this paper to see who got who got SARS-CoV-2, and by who I mean which wildlife might have SARS-CoV-2. Yeah.
yeah. So they set out some traps to capture the various rodents and mesocarnivores. That'd be like raccoons and other sorts, and they tested
Minks were
them
also
to see whether they had.
mesocarnivores.
Yeah, so they're not mega carnivores like your lions, your bears, but like kind of your foxes, your kind of um, coyotes, wolves. I'm trying to remember what American fauna are out there, and I just realized that my knowledge is not good enough. I was about to say, okay, and jackals, hyenas, and like,
And then
no, that's not.
cheetahs.
Yeah, but yeah, so they did some tests, and they think they might have seen some things, but they didn't really find solid evidence of SARS-CoV-2 establishment in wildlife yet. But I think that's a concern that out there, that if we do have SARS-CoV-2 become endemic, part of it will be due to wildlife being hosts
A
for it.
reservoir, yeah,
Yeah,
absolutely. Yeah, I think the reason why that this is not like they do RT-PCR, they don't like find live virus, right? I guess
right.
would be one of the gold standard, right? Sort of tests that we'd like to see. Like, are there active viral particles, right? Like, take some of that serum out, spin it down, put it over vero cells. Do we see plaques? If we see plaques, right, and then you genotype those plaques, and that's SARS-CoV-2, I think that that's like a really a more definite show of like infection in those places. Here, like they're doing RT-PCR, which I mean that's the gold standard in diagnostics for us, but we know that that only correlates to viral replication. It doesn't necessarily mean, yeah, that those viruses are replicating in there.
Yeah, so I mean, the mink tested positive for SARS-CoV-2 antibodies. I think some of them had like tested positive for some genes for SARS, but none had like antibody responses. So it could have been either they're exposed, but they just didn't get infected. Because I think we've seen before, like some animals just don't. So like mice don't generally get infected by SARS-CoV-2
Mm-hmm.
unless they're mutated. So we a couple of weeks ago we did see well, I say like it's nearly a year ago since we did our mouse models paper,
Mm-hmm.
where we looked at mouse models and found that there was a group of researchers who mutated SARS-CoV-2 to make it able to infect
Yeah.
mice, which wouldn't usually have it,
Mm-hmm.
and a lot of those mutations showed up in these newer strains.
Yeah,
So
yeah, and actually, like
that
when we were looking, I remember when we were looking through those papers, like it was funny to say, like, oh, we read this. It was like a model paper. Like, why are they referencing this mutation from this model? And it was because it was like experimentally found.
yeah. Uh, so yeah, I'm still kind of angry about those papers a little bit because I feel like when you do those gain of function experiments, you need to be very careful about it. But
Mm-hmm.
uh, because it takes only a short amount of logic. Someone goes, oh, they created this mutant in a lab, and these mutants have appeared, and then immediately, it's
For sure, yeah. But that's not the
so
case, right? Because we know like
definitely not the
there are
case.
mutations that come along with natural, like the route to natural mutation is this kind of like wavy path, and you can see like the different ways that it got there. So like when you put
Right.
those lab strains, I guess, on a phylogeny, you wouldn't see them associate with the ones that we find out there. It's just like what we were saying with the New York strain, actually, right? It's like
Yeah.
you can get to the same end goal of like more transmissible, right? That key mutation that got you there, but along the way, if you're doing it through the passaging route, either passaging in cells or in humans, you'll get like a whole bunch of other things that assort with it. They give it a character, a fingerprint.
Right. But anyway, that brings us to the next paper we're talking about. The B1.351 and P.1 variants extend SARS-CoV-2 host range to mice.
Yeah, it's
Um,
kind of full circle, right? So basically,
yeah.
I think I'm not sure if these mutations though were found in that paper, but I mean, one could, I guess you could hypothesize, right, that mutations that were that happen in the wild may have effects, right, to the lab models that we're using. And in this case, they're looking at the P1 variant, which is the Brazil, the Manaus one. I think that's how people call it. And then what's the other one? The 1351. So that's the South Africa
South
variant.
African.
Yeah.
Yeah.
And they put these in mice that are not modified in any way, right? They're just plain old laboratory mice, and they find that they get a disease.
Yeah. So unlike with the original strain just bounce off the mice, whereas these ones actually they cause infection, but they're not as deadly as the infections that were created by the mouse adapted strains we've seen in previous studies.
Hmm. Hmm.
So I mean, the interesting thing is like these are kind of an interesting new tool that people could use to study this disease because now like if you do a vaccine against these strains developed in mice, it actually is going to be representative of it more.
Sure.
Um,
Yeah, it gives us yeah, like now more people can do this potentially without having access to the same animal reagents. They do also compare with the B117, right? And that
yes.
doesn't seem to have the right mutation for this. So I think they're pulling strong towards a very specific mutation that's both present in the P1 and the B1, the South African and the Manaus strain. Uh,
Yeah.
that's where the evidence is kind of pulling us. And again, I think that it's nice that we're talking about it in context of these first three papers because I really think it's telling us a story of the virus going endemic potentially, right? Because seeing that some mutations that are occurring in the human population are making it easier to infect these small rodents, right? Might mean that then these are the strains that will end up forming the reservoir, right? That then like can bounce back into humans over periods of time. Um, yeah.
Yeah, so I mean, it could be like the way yellow fever primarily almost affects primates. So in order to check whether there's going to be a big yellow fever outbreak, people do have to like check primates in
Hmm.
the area where they've got yellow fever, like things like that. And the thing is, flu also we get flu coming from various animal sources as well. I mean,
Yeah, they do the
lots
monitoring of
of
populations and pig populations for flu, and that's I, I don't know. I actually feel like again these first three papers to me it shows like us as humans have done a bad job of controlling this outbreak. And I mean, of course, we were relatively unprepared. Didn't do such a good job in controlling it, so that there are now many ways for this virus to find a new home in the world. And in doing so, it just may be very difficult to eradicate potentially.
yeah. So I think we'll be having because I mean at this point looking at it, it almost seems like it's very difficult having the idea of eradicating it in the first place might have been very ambitious, because this is like one of those common cold viruses now, or like the flu,
Mhm.
where except with is a bit more deadlier. So the question isn't so much how we can eradicate it, but how we can reduce deaths and how we can survive it.
Absolutely, yeah, and again, like maybe the future is not about like no SARS-CoV-2 in the world, but some sort of management strategy, right? A combination of surveillance and vaccination that gives us like some sort of control over the scenario.
Yeah, but one thing that are giving us some control of this scenario is vaccines. So let's
Ah,
talk about because we've got some good news about vaccines actually like doing their jobs and
yeah,
working.
absolutely. I think that I'm so excited that vaccines are out, right? Because it does right from we were reading those clinical trials, right? It's showing us this great efficacy, lowering the amounts of infections, the symptomatic infections, lowering the amounts of people that go into the hospital, right, for serious disease. That is enough to convince all the regulatory bodies that we're ready to get it. Many countries have had their vaccine rollouts. But there was that nagging question in the back of my head that I was bringing up constantly, that but what about asymptomatic transmission, right? Like, we'll get these vaccines, but we're still going to be able to spread it to each other, and that might not be a good thing. So these studies are not randomized control studies in all of these cases. I think that they're mostly either longitudinal, like cohorts, maybe some perspective, some observational. But the idea is that with so many more people getting the vaccine now, we can start figuring out, like, from these large sets of data, some other characteristics, beneficial characteristics of the vaccine. And importantly to me, I want to see transmission rates.
Right. So the first one of these, so you've got 4 papers that are talking about how the vaccines are working quite well. And so the first one we got on our docket is the effect of vaccination on transmission of COVID-19, an observational study in healthcare workers and their households.
Mm-hmm.
So this,
So I think
yeah,
it's
this,
definitely off the back of the idea that healthcare workers are some of the first people to receive vaccines. That's why we're seeing this data come out now, and it's like focused on this one cohort. And like, you can imagine that when we're looking at healthcare workers, there's going to be some caveats we should think about, right? Because those people are probably exposed more to SARS-CoV-2, which I guess is great. They're being challenged, right, in a very real world setting. But they also, right, like, they're also not necessarily like the sickest people, and right, like, there are features of this population that are not representative of the whole population as a whole.
right. They tend to be
Um,
healthier, younger. They have like jobs there. Tend to be skewed like to more women working in like nursing and roles like that as well. So there's a lot of different bits in healthcare workers that are slightly different for population, but they're a good indicator.
yeah, I
So
mean,
this and
they're a large human population, right? There are definitely some things that they are representative of. Okay, yeah, sorry, go ahead.
and this is a good one because they just look at the healthcare workers. It looks at their households. So it looks at like the healthcare workers and the probability of them spreading it to the rest of the people in their house. So if like say someone gets infected and they go home, how likely is it for them to transmit to their family members? And
Hmm.
how does the vaccination change that?
Yep.
Um,
I really like that approach where they like focus on the household members, right? Because it gives them this control over the data set, right? Where it's like they're really looking specifically at this one relationship link, right? And of
mm,
course, those household members, they're also mixing with others, so like that's like one of the confounding factors. But like, they tried. This is the difficulty of these data sets,
mm,
retrospective data sets, right? Is that like you're trying to get close to the scenario that you're measuring.
yeah. And they do find I think a 60% reduction in cases in healthcare workers who have been vaccinated, which
Hmm.
is a pretty big deal, I think, in terms of like reducing the spread of the virus.
Yeah,
Um,
absolutely. And I think that is more pronounced versus from the first dose to the second dose, right? Like, full vaccination is helping improve this parameter a little bit.
yeah, I mean, one of the sentences I pulled out of this paper that I thought was quite interesting was there were no hospitalizations in healthcare workers who received a second dose. Therefore, we couldn't calculate any data from them. So that was kind of that. So that
Well,
was
that's
kind
like
of
that's just another, like, we've seen that in the clinical set data, right? Like, that's like
yeah.
this vaccine is good about uh, um, stopping us from getting serious disease, right? That's why we're excited to get it, right? Because I don't
Right.
want serious disease.
Yep. So next one is the impact of COVID-19 vaccine on asymptomatic infection among patients undergoing pre-procedural COVID-19 molecular screening.
Yeah.
So these days, whenever you're going into hospital and you're going to surgery or any sort of medical procedure, they do swab you to test for whether you have COVID-19, and this is a test for of those people because they aren't coming in for symptoms; they're coming in for stuff that's completely different, and
Yeah.
so this will give you an idea about asymptomatic transmission or
Yep.
infection.
Yeah, yeah, and I think this is, like, again, very exciting that they're able to get this data together. you can imagine that, probably this group is a little bit sicker than normal, but there's also
Hmm.
elective surgeries that you'd come in to get at the hospitals, right? And like that's being lumped into this group as well. So yeah, like that's the population we're looking at, not healthcare workers. So it's kind of right. This is shoring up our story, right? Like as we add different populations into the mix, we get a better idea of like what exactly the vaccine is preventing. And these people probably aren't at high. They're not always seeing SARS-CoV-2, right? So it's more similar to that scenario. And once again, we're seeing really good reductions of risk of asymptomatic SARS-CoV-2, right? Like I think
Yeah.
looking at things like, again, better after the second dose than the first dose, but there's still effect even with the first dose. I think they have these unadjusted relative risks, is how they're reporting it.
Mm.
So, one would be like the highest amount, right, of risk. And then as you get lower, right, like that's a lower amount of risk. And you're seeing like maybe like 80, right? Zero point 0.2 is 0.28 is some of the risk that we're seeing. 0.4, so that's like 80%, 60% reduction in the
Yeah.
probability of having SARS-CoV-2.
So yeah, that's pretty good news. Let's see what else we have. We got decreased SARS-CoV-2 viral load following vaccination.
Yeah, so I picked up a lot of these papers from a CDC website. They were bringing it together, and they were trying to explain this whole issue of like, right, like what is asymptomatic transfer? I mean, hopefully we're thinking about that right now. What is asymptomatic transfer, and like how are the vaccines affecting that? So this is kind of like a hint to the mechanism, maybe, right? Of like what would make asymptomatic transfer lower. And the fact is that if you have less SARS-CoV-2 in your body, right, then you probably will not transfer it as much. And so here we're seeing a little bit a more detailed look, right, into viral load in individuals who have been vaccinated.
Yeah, so they basically do RT-qPCR measurements on people who are going in and out of this laboratory, and they're just and they got the count values, I think, or
Yep, the CT value.
cycle threshold. Yeah,
Mm.
that's I keep thinking of count, but anyway, it's cycle threshold values, and so they actually literally measure the amount of SARS-CoV-2 RNA in people, and they notice that people who are vaccinated, like if they do have it, it's much lower.
Yeah,
Um, so it's not like they're like sampling a whole bunch of people who don't have the virus, and then the rare people who do.
yeah.
They're like looking at just the people who have the virus, and they have things that are lower. So that's
Mm.
quite a good sign.
Yeah, absolutely. It's again, it dovetails nicely like as a reason for why there's less transmission. And again, I think maybe I should reiterate because like it's easy to get wrapped up in, to me, the amazing news of all of this. Like it is still possible to get it, right? It is still
Mm,
possible to spread it, but the likelihood is that much lower. So if we go back to that Swiss cheese model that we're looking at, right, we can just know that this vaccine cheese layer is pretty good, and like we should be applying it widely, I think is what I'm reading here and what is getting me excited about this like little bolus of data.
mm. Yeah, that vaccine cheese layer. Um, sorry, I love that. I'm gonna get my vaccine next week, so I look forward to getting my vaccine cheese
Cheese
layer.
layer, yeah, just this like force field of cheese.
Yeah, yeah, delicious. So next is the interim estimates of vaccine effectiveness of BNT162b2 and mRNA-1273 COVID-19 vaccines in preventing SARS-CoV-2 infection among healthcare personnel, first responders, and other essential and frontline workers. 8 US locations, December 20 to March 2021.
So that's a classic MMWR like title, right? Like this
Yes.
publication is it's done by the CDC. It's always about like the cutting edge of like what infections are out there in the world. Of course, SARS-CoV-2 is making up a big part of it these days. And it's not written for, like that title is written for epidemiologists, for doctors, right? It's like giving them the update of what's going on. But it's great. I think it's
Yeah.
like very without actually it doesn't say anything about what the interim effectiveness is. It just tells you like what the group is. But if you dive into the
Yeah.
paper, they're finding a favorable reduction in patient years, I think, is how they're measuring this, who are infected. Yes. 82% in some cases, right?
Yeah.
90% fully immunized.
And the thing about this is separates from the other studies. It's a prospective study, so they actually looked at people who healthcare personnel, first of all, all these people who were about to get vaccinated, and they set them up in these groups before, and then they looked at followed them around. So it's a lot. So it's a lot more stronger in that way.
Yes, yeah,
Um,
stronger.
so
Hmm.
they've
Yeah,
got like
because
this thing called the Heroes Recovery Network, which is a longitudinal cohorts in all these different locations that they are just tracking to see what's happening to them. So they already know like kind of who's in it already,
mhm.
and they're seeing what happens.
Yeah, so I guess yeah, what makes it stronger is the fact that they don't have to guess like for each individual that they're following, they know the exact date in which that they've gotten their vaccination, and then they can make sure that all of those I guess data columns are lined up properly. But in retrospective ones, sometimes you're asking people, sometimes you're pulling that data from like different sources. So like that's what sort of makes some of the earlier studies a bit worse. Again, this is just healthcare workers and people who are already, but actually, in my mind, it makes it even more convincing because these people are in scenarios where they're going to be exposed a lot. What we see here is like, oh, one thing that might be a big confounder is that they really use PPE very well,
Mm.
right? So, like,
Yeah,
again, but to me, again, it's just flowing into a great narrative. Use your act like these people, right? Like, get your vaccine, use your PPE, and you can expect, right, that your rate of transmission, the possibility that people will transmit the virus is way down. And, it's, again, very exciting news that just shows us that the tools that we have out there can be very, very effective for controlling the spread. But again, it requires more, in addition to getting the vaccine, also practicing the proper behaviors to not be exposed.
yeah, and that brings us to the end of the good news part of the vaccine segment. The next part is going to be a bit of a difficult one because we're going to be talking about the vaccine side effects. So there's this story that's coming out in the media quite recently about this thrombocytopenia like side effect, and I think the thing about talking about so I mean, when we started the phase 3 trials, if we recall, they were for about 30,000 ish people.
Mm-hmm.
So in those side effects, you kind of sorry. In those studies, you'd expect to see side effects appear like 1 out of every 30,000. But now we're getting to the point where we're vaccinating millions of people. Now even rarer side effects are starting to appear that we could not. And so I think the point I'm trying to make is no matter what side effects we find, the magnitude of the side effect is still not going to be bigger than the magnitude of the threat of dying of the disease still.
Mm-hmm.
So I think that needs to be kept in perspective. But
Absolutely.
there are, but that leads us to one of the complications of like mass vaccination campaigns and also the precautionary principle for when it comes to dealing with vaccine side effects. Because when you have a normal drug and you find a side effect, and it's a drug for say something that is a lifestyle condition, like if you're dealing with something psychiatric or something, then halting the drug is the sensible decision
Mm-hmm.
because halting the drug you're preventing harm whilst you figure it out.
Mm-hmm.
Whereas with vaccines, that precautionary principle is almost turned completely on its head because if you don't vaccinate people in the middle of a pandemic, people will die that could be prevented.
Mm-hmm.
But and you have to weigh that against the people who die because of the treatment, and so you're going to see almost so if you want to see the most save the most lives, and you see a rare side effect, what do you do when you have a vaccine? I mean,
Yeah.
uh,
Yeah, I think in general, like we usually anchor those decisions also on the phase 3 trials, right? Like the side effects that come out from the phase 3 trials, there were side effects, right, that came out, and they weren't serious, right? None of them were serious, I think. But that's where we anchored that value assessment that like we're willing to see this much, we're getting it out there, right? These many this efficacy for this adverse events profile, and we made the decision already, right? We are giving the vaccine out
mm,
in massive amounts, and as you say, like it's a judgment call, right, that I think is in a one that is like necessary to be made because there's this public health at play, and if we were
mm,
if people were being very conservative about this information, right, and saying like, but we want to see no side effects, you would end up with we'd be back where we started, right? Like we've cleared a lot of different boundaries, right? Like these are extremely rare side effects. I think is just the important thing to highlight.
Yeah, and also the big concern when you're vaccinating any large population, when you're doing any treatment large scale, you have to realize that when you cut any population in half, there'll be some people with a disease that'll go in one half, they'll be more prevalent in one half than the other, just because of random chance.
Mm.
Random assortment will mean that
Hmm.
some things. So you have to be able to take get get past all that noise, all that kind of background disease to figure out whether it's real or not, and that's a real challenge of trying to of this sort of case.
Hmm.
But um,
So
and
yeah, what's this
also,
first one? This is a report of the serious effects that were found.
yeah, yeah. So this is the actual. So this is probably the only report I've been able to find that actually goes into in depth into trying to not only just figure out what's happening, but also figure out how to treat these cases when they occur. So,
Hmm.
so this actually looks at the laboratory features of 9 patients in Germany and Austria who developed this thrombosis and thrombocytopenia event.
Mm.
Um, and what they found was that that quite so they had like one in first, so it starts off with like it's a case report, so there aren't any control groups,
Hmm.
which you kind of would have. I mean, at this point, given the rarity of the side effect, this is all we're going to have for the most part, because it's going
Hmm.
to take a long time for anybody to get enough information about this. So what we have at the moment is just a few case reports. They are kind of like anecdotes, but we can still learn something from them.
Yeah.
um
I think maybe to help people, like, because I think that's an interesting point that this particular paper is just about these certain events,
mm,
right? But it's not set up to show us like the frequency of the events that happen. And actually, but we can infer the frequency, right? We can just say out of everybody who's been vaccinated in these populations, right, is the frequency of them. Yeah, so there's
yeah.
quite a large denominator on these.
Yeah, and the thing is, like there's been about like 30 cases of this recorded in Europe. So 9 patients would actually be quite representative of that those 30 people who are infected, because it's still a very small number of people. So even 9 patients is about 30% of what's been discovered so far.
Yeah.
Um,
Do you know how many people have been vaccinated in Europe at this point?
well, I mean, according to like a couple of papers I read, this is about out of 5 million.
Out of
Um,
5 million. Yeah, okay.
Yeah, so this is out of 5 million, and since then the more people are vaccinated. But again, once people get better at recognizing these cases, we might see a lot more of them being spotted earlier,
Mm.
because at the moment we're seeing those 30 people are the fatalities. But
Hmm.
actually, people might be feeling these sorts of symptoms more commonly, but not, but they never turn into anything severe, or
Mm.
they so it's kind of difficult to, because
Yeah,
then you've got that background level of disease that you need to take be able to distinguish things from.
right.
Um,
And so this paper in some ways is helping, right, is for doctors, right, to be able to
right.
make some clinical, like to gain some clinical insight into what went down in these particular patients so that if they see it in their own region, right, and also for maybe policymakers, right, when they can maybe create a definition to say, oh, we might be able to deal with all of these things with better management, with better, yeah, medical
Yeah.
management.
So what they found, they when they looked at the serum symptoms on these patients, they tested them for antibodies against heparin uh, and also for the antibodies, I think, to sorry, how platelets react to the presence of heparin and to the coronavirus vaccine, I think. So they, so this is interesting. So they tested the patients who showed up with the disease, and they also tested their controls were people who were also vaccinated with the same vaccine, but didn't show any symptoms.
Mm.
And so the same reactions, these patients had their platelets activate in response to this, but patients who were also vaccinated, but so this is going to be difficult for me. I'm finding it quite difficult to explain this because my brain's a bit on the boil about this. But uh, so some of the people who had the vaccine who turned out with severe symptoms
Hmm.
are reacting to in this disease called like uh, uh
Thrombocytopenia.
yeah, thrombocytopenia. So this is what's going to be called like heparin induced thrombocytopenia.
Hmm.
So um so heparin is usually an anticoagulant, so
Yeah.
it's produced normally by the human body, I believe. And and but so usually it's given to people to do the opposite of coagulation. So,
Hmm.
but sometimes there are some people sensitized to it. So then that causes actually the opposite effect. So when someone is given heparin, this disease happens, causes a drop in platelets because their platelets are forming clots,
Hmm.
and then lead to all these like nasty outcomes where clots form in blood vessels that lead to infarctions and other sorts of nasty diseases.
Hmm.
Um,
Yeah, so
so
in these particular cases, so in some ways that could even be the signal, right? That people could look for, right? You could try to see the like if people are hmm, you'd have to be able to segment, or you'd have to be able to segment the population to see, well, who is at risk actually of going down this path, and then they provide a diagnostic that you could do, right? If they have autoantibodies towards this type of heparin, then maybe they need closer attention. Maybe they should go into the hospital afterwards. Um,
yeah,
and again, it's
so
not a very common thing, right? I think that that's the important part. It's like, uh, do they say comorbidities of the people in this group?
yeah, so two of these patients had pre-existing autoimmune diseases.
Okay.
Two of them had underlying coagulation disorders. So so
Right.
that so basically around just under half already had an underlying clotting disease
Hmm.
or something that be used to could be a, a covariate that could have made them more vulnerable to this sort of disorder.
Right.
Um,
Yeah, and again, that's like it's hard because they haven't done like that study to do the full association, but it again, from the clinical perspective, it provides tools for doctors to say, and maybe even tools for patients in some ways. I guess that's not what the news has been uh, selling to us, but that idea that if you have these particular pre-existing conditions, right, maybe this is something you talk to your doctor about, or like, right, this
yeah,
is, yeah, it's not for everybody in the population that this risk exists.
and the complexity here is that some of these patient populations would be like moved up the vaccination order because like having a coagulation disease it actually means that you're more vulnerable to COVID because
Hmm.
COVID causes those. So again, so there's been a lot of talk about affecting younger populations, but there are two types of people who are getting this vaccine as younger. Usually, healthcare workers or people with these underlying diseases who might be
Hmm.
vulnerable to this sort of thing. So uh,
Oh yeah, so it might even be less common than right, it may be even less common than we think because of the way the vaccines were rolled out.
yeah, or
Hmm.
I mean, that's the problem with having such a small study like this is that it might not even be related because you're if you pick any set of patients, you're likely to find several underlying disorders just
Sure.
because you're selecting from this population. So it's very hard to actually pull out anything that's generalizable from this. Um,
Yeah.
uh,
So I guess
but
at the
what?
end, like what I'm taking from this is it's very frustrating to see the news
Yes,
focus on it because of like what limited information it's based off. The narrative is based off of.
and the one thing that news haven't been really talking about that I think is probably the most important part of this paper is that all these reactions were blocked by monoclonal antibody IV3 and IVIG. So they have in the study they've
Therapeutic.
tested out a treatment that could be used to stop this from happening.
Hmm.
And so the next stage of this would be like if someone feels the symptoms like this, and these kind of cytopenia, if you go to your doctor like almost like immediately the moment you find symptoms and they know to look for this, they can actually get you a treatment, but quite
Hmm.
early on. So the next stage of this would be to see whether like how they treat this because I mean vaccines do have side effects like so the reason so when you have like your flu vaccine or anything they ask you to stick around for about 15 minutes afterwards because that's about the amount of time it takes for a really severe on like anaphylactic shock to set in. So they wait for that so they can because they can be treated very quickly, but they just have to make sure they want to keep you around. I feel like it's almost the same thing like this where if there's something you can just go to your doctor and then they can provide a cure. So
Yeah, absolutely. Yeah. And again, I think maybe like the most instructive thing for me is that because it coassorts so strongly with these comorbidities, then it also just like, this is good information to get out there for people who might want to bring something up to their doctors. Um, but yeah, but
but
I think not a very common thing event in any case.
no, it's not a very common event, but it's something to keep an eye out for because while we don't want to like make too much of it, we don't want to like cause fear about it. It's just I think raising awareness that it's there that
Hmm.
is kind of important for the medical community, so they know how to deal with this sort of thing when it appears. But
Hmm.
then
Hmm.
again, there is a strong like so I mean there is a strong element of calling for shouting fire in a theater, kind of to the news coverage of this,
Yeah.
which is something that also makes it very hard to talk about these side effects, because the moment you say something about that, I mean, vaccine hesitancy also kills, and so if we talk, I know I'm breaking this rule because I'm talking about this on this program, but if we talk too much about it, the fearmongers are always watching. So
Yeah.
I mean, like early on in this outbreak, there were a couple of people who were falsely saying that masks did not work in a misguided effort to try and stop masks to stop a run on limited stocks for masks, and
Yeah.
that's fed into an anti-mask sentiment that's
Hmm.
still persisting.
Hmm.
So imagine what will happen if people do the same thing about vaccines.
Yeah,
Um,
exactly. So yeah, I mean, again, I think that it kind of touches on that frustrating element, right? That it's like it's nice that there's news coverage about it. People do need to know, but the news needs to dig a bit deeper, maybe. And I think that like I didn't even know that like yeah that there was a therapeutic proposed in the same breath, right? As telling about these adverse events.
yeah. Um, So the next paper is the only one that was I could actually find that was published on this, which is a shame, is because it's a correspondence letter, so it's not actually proper research. It's just some researchers have put together some modeling
Hmm.
and some data. So they're looking at, so the title of this paper is Thromboembolism and the Oxford AstraZeneca COVID-19 Vaccine Side Effect or Coincidence. So this paper attempts to put all these like vaccine side effects in context of saying like, okay, are they actually more greater or lower than the population? Um,
Hmm.
so I mean, what they find is that they they pull in a bunch of records from the Danish repository. So there's a Danish healthcare system has a repository of like how of diseases that happen and how often they happen.
Hmm.
So so with the idea of like we can figure out what the incidence rate just normally in the population, we can figure out, okay, are these vaccine rates actually higher or lower than what we'd expect?
Hmm.
Um, and so they say it's like in a population of 5 million people, the size matching the approximate number of people who are having received the Oxford AstraZeneca COVID-19 vaccine in Europe by March 10th. This incidence would correspond approximately to 169 expected cases of venous thromboembolism per week, or 736 expected cases per month. Uh, so we
Oh,
haven't
that's
really
quite a
seen,
bit. Yeah, it's quite a bit.
yeah, that's quite a bit. So we haven't really seen that amount coming out, but again, data takes some time to come to shake out.
Hmm.
Um,
Well,
so
no, but I mean, this is the Danish group. The Danish data here is being used to find like this is just the amount you would see in this in a population of people, regardless of what was going on in the world, right? Like this is
right,
like yeah, like just taking like the rates of known disease, and then like if it really was that SARS that the vaccine was having this big effect, you'd see some sort of bump in them, right? Like that's what you're looking for in the as the signal. But I mean, already just from hearing the previous one, it doesn't like there's so few cases that even have a loose association, so the bump isn't happening.
yeah. I mean, it's yeah. I think it's like there is so few cases that I mean, I'm not saying that the case because I feel like this almost goes too far in the other direction to minimize the if
Yeah.
because
Well, because the other thing is that we're not. I guess what we're missing, right, is that like the missing link between the two papers that would help us understand a bit better is just like um, in all the cases that happen of venous thromboembolism, what are the different reasons that we could say that are associated with it? And then
yeah.
maybe we could understand a bit more about like what it means to have these cases appear in the context of the vaccine, right? Because the idea is that like it's happening in the background anyways, right? For many different reasons. How do we know that the reason is because the vaccine is being given?
Yeah.
Mm-hmm.
Um, yeah, so this is going to be an interesting debate going forward because again, we're trying to make decisions on very incomplete information. Um, and I had a point I was going to make, but I've
Oh, I'm
completely
sorry.
blanked on it. No, no, it's fine. I'm going to go through my notes to see whether I wrote it down. Um, uh, no, it's completely gone. No, never mind. We'll probably end up talking about because there will be more studies on this on ongoing. So
For
I feel like
sure.
we will be talking about this for a while. I mean, I imagine this will be like a talking point about the vaccine. I think there is still that. I mean, because they don't actually focus in on the specific heparin induced thrombocyte
Yep.
thrombotic disease.
Yep.
And I feel
This
that's
is
an
just
important thing.
thromboembolism. It's like
Yeah,
it does. I think it's again, it's a more general term, right, than
yeah.
the specific one, and so that's why not really related. But I mean, no, it is related, but it doesn't give us enough detail to really like tie the two stories together. Because we're not very good at knowing what side like, we're not really
Yeah.
medical doctors, right? I think that that's like part of the struggle here and part of the struggle for everybody. No one's really the medical doctor to be able to put that in context in an easy way to understand. It's not easy for us either.
Yeah. Um, so I think that brings us to the end of this story basically. I'm going to be getting my vaccine next week, so I'll be on the lookout in case I might get the AstraZeneca vaccine just to see whether I see any symptoms or not. So the things I do for the clicks and views, please subscribe to.
Okay, and we have a few more SARS-CoV-2, but moving away from the side effects topic, this is maybe more in line of some of the basic research that we've been seeing coming out. SARS-CoV-2 infection of the oral cavity and saliva. Um, we've looked at a bunch of papers that have talked about the different tissues that this virus can infect, and this is basically one more one more data point on that long list. They do single cell sequencing here, so they're getting some really specific resolution on what cells in the mouth might support SARS-CoV-2 replication, um, and yeah, I don't know what to say. I
Yeah,
really like all the pictures in this particular paper. They do a good job of contextualizing where their immunohistochemistry is coming from.
yes, there's some really cool. Wait, is this one? There's one that's had one of these papers we're going to look at has really cool technology. I know, but this one is quite cool because they've tried to profile all the cells to see which ones are most likely to get infected with SARS-CoV-2. So
Mm,
which ones are expressing TMPR, SS2, TMPR, various like, and also ACE2 and
yeah.
various other proteases that could potentially help SARS-CoV-2 gain entry.
Mm.
And then they do droplet digital polymerase chain reaction, which is something I haven't really heard of before, so that's an interesting thing. Um,
It's the same thing as all the single cell stuff, actually, right? Like
yeah,
it's like you make sure that the thing that you're no, this is the same technique as all single cell stuff. Is that like
right,
because you have to isolate all these cells in their own reaction to amplify the RNA, and it's usually done through this oil droplet style.
right, okay, and yeah, so yeah, I see, yeah, that's so yeah, this is another like one of those single cell atlases. This is done in humans, and it's done in an area that is probably quite important for.
I don't want to minimize the cool factor of single cell. It's just that now that we've seen a few, like, because it's like we're gonna see it a lot. I'm not gonna ooh and ah every time it comes out, but it is a very powerful. It is a very powerful technique, right? Because you're getting this resolution of like we know that this expression factor is happening here, or like this thing that we're looking for is happening in these specific cells.
Yeah, and I think this also tries to like look at the timing of when different parts of the oral cavity get infected. So it's one
Mm,
way to try and explain why false negative tests can happen because of where the SARS-CoV-2 is during that period of infection. So I
yeah.
think that's quite an interesting thing to look
Mm.
at.
Oh,
Um,
I think also what this paper is kind of nice in closing the loop on the story is that we've heard maybe people have heard about there's salivary tests, right? Like where you can spit and they can figure out if you have SARS-CoV-2 or not. This is actually a really nice story to close that loop. Actually,
yeah,
it's not like that's not weird, right? It's not that like oh it's in your blood and then somehow it gets there. Actually, no, it infects some of the tissues that make the saliva and you know are bathed in saliva.
right. Next up is kind of adjacent to that spatial landscape of lung pathology during COVID-19 progression.
Hmm.
Um, So this is interesting because it like looks at 23 patients. Like they compares like COVID-19 respiratory distress syndrome to post-influenza respiratory distress syndrome and bacterial pneumonia and other kind of diseases. So
Yeah,
that's quite interesting.
yeah. Trying to see if I guess there's something unique to the particular infection that SARS-CoV-2 provides. really very colorful pictures.
Yes. This has got imaging. This is the one I was quite excited about because imaging mass cytometry, which is based on like they add an antibody that's labeled as a metal, and they add it to some immune slides, and
Hmm.
so that will just target like a bunch of biomarkers. They also do targeted transcriptomics where they like cut out certain parts of like a slide and they sequence all the RNA in that, so you can actually get an idea spatially of where the RNA is in the body. So that's
Yeah, this is a
so
very high level of detail, right? Applied to, on the tissue scale. I mean, that's why they call it spatial landscape, right? Because, in general, when we think about biology, like you do a lot of crushing of the tissues in
mm,
tubes, right? And then you get an average of everything that's happening in that region. But as technology has gotten a lot better, people have gotten a lot more specific about things, and this is one of those. Incarnations where you can now just go in and you can see, oh, in this slice that I've taken, right, I can tell you what proteins are there, what transcription is going on there, and then again, they're building a profile of SARS-CoV-2 infection versus some other respiratory infections.
yeah, and they do it's more that like relationship mapping where they look at how single cells like predict related to each other based on their genes, and so there's a
Hmm.
lot of like cool like data analysis stuff they're doing in here as well.
Hmm.
Uh,
Yeah, this is like a I feel like a lot of science is going down this path, right? Like to try to zoom out more. Like people have zoomed in so much that like now I want to zoom out and I want to see like with again as you zoom out, like it matters how big the pixels are, but like you're getting a really different picture of what's going on. You can see patterns that you weren't able to see before.
yeah. Now we're going to move to the next paper, which is sequelae in adults at 6 months after COVID-19 infection.
Yeah, there's not a lot of information, not a lot of published papers about long COVID symptoms, right? I was actually reading a review that was someone was arguing there needs to be more patient advocacy, right? Like just people aren't paying attention, and I guess it's a complicated thing because there is this very acute need to deal with acute infection and make sure that those people don't have bad outcomes, but there is also the potential for this long-term effects. And it seems like it's fairly well accepted that there are some SARS-CoV-2 long-term effects. Not really sure who it affects, right? What populations are most susceptible to this? I think that's sort of unknown. But in this one, they're taking, I think they take like a quality of life metric, right? Like,
Mm.
can you do your day-to-day activities well? And they show that for some people, you can have symptoms that affect your day-to-day life up to 6 months afterwards.
Yeah, these are very difficult studies to run because questionnaires you need to be very careful what questions so often most of these will use like standardized psychiatric questionnaires that they've
Yeah.
spent a lot of time and effort trying to make sure phrase the questions in a certain way and they're standardized so they can be compared with other studies.
Hmm.
Um, so
Yeah, and then it's
uh,
a really hard signal to find because like that's a lot of intervening time between like the proposed event that you're associating with these symptoms and anything else. So I think that's another challenge. But
yeah,
I could see, I mean, from this paper at least, they seem to think that this is the paper that's often cited as the one where it says fatigue is the thing that's associated strongly. And they sort of expand in it a little bit more with a host of other COVID-19 symptoms.
yeah, this is good because it is a longitudinal prospective cohort of adults. So this that's actually really good. Um, because this is the kind of study I'd want to see for long COVID, where they set some people in advance and then they follow them to see what happens to them, rather than taking a barrel full of people who have long COVID and then looking back at the history. I feel like this one gives you a much wider viewpoint on the actual
Absolutely,
prevalence of it.
absolutely. Yeah, they're able to, and also in terms of like as they follow, they can drop the data points as needed, right? If they see confounders, so that also gives them control over understanding the associations.
Yeah, so I think that because yeah, you're absolutely right. We don't have enough about long COVID, and we really do need a lot more about that. Uh,
Yeah, I think that
but
that's for some people, I think it's a motivation to get vaccinated and make sure you're practicing your social distancing, right? Just to know that there's the specter of this out there. But I think also from a very practical point of view, like so many people are being infected. It would be nice to know like the burden on the healthcare system and different ways that we have to the healthcare system will have to adapt to deal with some of these potential symptoms.
yeah, for sure. And I think now we've got finished the COVID era of our segment, and we can now talk because now we're expanding. We're talking about other microbiology bits. So
Mm.
the next paper we're going to be talking about is swarming bacteria undergo localized dynamic phase transmission in order to form stress-induced biofilms. So this is this is essentially this paper is when bacteria behave like multicellular organisms, and why do they behave like them? So like bacteria, they can form biofilms like the plaque that forms on your teeth, or sometimes they
Mhm.
can form swarms where they kind of link together and they behave almost like as a group.
Mhm.
Um, and this paper aims to try and figure out why they do that. And part of the thing that like made me interested in this paper is that it has lots of like cool little videos, which are very, of
Oh,
like this of
bacteria
bacteria.
living around.
Yeah, this is why I like eLife because they actually like try to present the data in a way that is like understandable.
Sure.
So I
Yeah.
so
There's
even if we don't,
science advocates, right, at eLife, and I think showing data in different forms is a part of that because and I think embracing the digital nature of publication is also a thing that doesn't happen always, right? Because you get trapped in like, oh, actually, this figure has to be in black and white, or it costs more money to publish.
oh gosh, yeah. I mean, I like and most people, most journals try to hide away the supplemental figures, whereas eLife actually was one of the first to make that effort to show it alongside the normal like figures.
The
So
norm. And I think they also one of the things that is a nice thing as doing like one of these journal clubs is you can see the reviews, I think, or they've been piloting. You get to see some
yeah,
review reports coming out from the different papers.
yeah, you can find the decision letter and the author responses. So yeah, this is kind of a cool paper. So I'm just showing up some of the videos of just it shows how these bacteria spread and how they react to like an antibiotic and how they kind of form biofilms around the antibiotic almost to try and so this kind of does some modeling looking into that and why the bacteria do that.
Hmm.
Um, so I'm
Very
a
cool.
I'm a
I
real
love.
sucker for videos.
Yeah, I really enjoy bacterial behavior, right? Because it's so simple to think about. I feel like as someone who loves bacteria, like I know that they have all these behaviors and I want to know more about all of them. But I think it's a great thing to show to others because maybe when you think of bacteria, you think, oh yeah, they grow or they don't grow, right? Like,
Right.
but actually, they do a lot more because they live in complex fluid dynamic environments, right? And actually, they also live usually in not very concentrated, right? They're not very
Mm.
concentrated when they live together. And so that's a very interesting thing as well that, like actually what happens in a small cluster of bacteria is probably more like real life than how we do it in the lab.
Yeah, and it's also interesting to think about like how they actually coordinate because they don't actually have many ways to communicate. They have various chemical signals, and how they communicate and coordinate to do mass behavior.
Yep.
Uh, so and that
I
brings
think
us
that's a
to
nice segue into our next paper.
yes, that's right. I was doing my segue into the next paper, which is all about taking advantage of that bacterial communications in order to get them to work for us.
Hmm.
Um,
Yeah, this paper is to me in one of the holy grails of microbiota research, right? Like the holy
hmm.
grail application is that if we understood these relationships between bacteria well enough, we might be able to introduce a bacteria into our microbiota that just gives us the best microbiota ever.
Yeah. So in this case, they looking at this from bioengineering perspective, saying like, okay, in it, we've kind of perfected the idea of creating a single monoculture of bacteria.
Hmm.
But what if we wanted to do something more complicated? So a couple of weeks ago, we're talking a lot about syntrophy and
Hmm.
uh, the, the 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 complexity of various bacterial communities that work together. So in this
Yep.
case, they actually tried to
Engineer it.
implement ways for to bioengineer bacteria to coexist with each other.
Hmm.
Um, And they essentially have like this the strategy of they have one bacteria that produces an antibiotic, effectively a bactericin, which
Hmm.
bacteria use to fight other bacteria for space. Um, and so this will secrete a bactericin that will kill off other bacteria.
Hmm.
But in order to get it to coordinate, what they have is another. They have this like the control of that bactericin put under this promoter that is only activated in the presence of a bacterial pheromone that is
Mm.
produced by another bacteria. So the basic idea is that there are two bacteria. One is producing this pheromone, and when it's in a very high concentration, it will actually cause the other bacteria to produce an antibiotic to kill it off. And then
Hmm.
once it goes below a certain concentration, that one bacteria will stop producing that bactericin, and then there'd be basically essentially be in a balance of where if one gets too high, the other one will kill it off, and then the other one will crawl back up again.
Yeah.
Um,
It's a very engineering way to think about it. Yeah, sorry, I was like, oh, this is like dropping it into a naive microbiota. It's actually building the microbiota from the ground up, right? It's
yeah,
like what goes into like making sure that we could get the strains that we want all together, right? In a certain proportion that gives us an effect. Um, yeah, and you can imagine actually that's useful for more than just health, right? Like this could even be like for manufacturing, right? Like, what if you're making things with bacteria and you need like a dash of this and like a bunch of this, but you don't want to have to measure that out, right? You want that to be evenly distributed over a surface, right? And you want the bacteria to get to the right proportions. This is the system that you could employ.
yeah. And let's say you want to modularize. So if you're creating a chemical that's producing, so let's say you have one bacteria that can do one chemical process, another can do another one, or you
Mm.
have various different options for a
Yep.
product. So in that case, you can change the chemicals not by genetically engineering the bacteria, but mixing in another bacteria with it that does that second step of the process. And so using these communities, you can, so, but that basically almost creating them as Lego bricks that can hitch together and do different parts of the reaction. So you got one that say converts carbon into sugar, and then it produces all these sugars, and then you have different options of what you can do with those depending on what other bacteria you mix into it. So you get this kit and just mix them together, and you create this community that can eventually produce that chemical you want.
Yeah. Yeah, so I mean, this paper has nothing to do with those applications we just
Nope.
spouted
Nope.
up. It's sort of the advance that would let us get to there potentially if they could make it work, and if they can make it work repeatably. And of course, this is also like the bench, so then like it matters like scaling that into whatever potential application. But very cool stuff.
Yes. So the next paper has a little bit of that bioengineering thing in it, but so the prophage and plasmid mobilome as a likely driver for bacterium abscessus diversity.
Yeah.
Um,
So I think this is more about to me this is like a more like an ecology kind of thing, right?
right.
We're looking at we talked about antimicrobial resistance in our previous deep dive, and it's sort of well known um, that antimicrobial resistance can get shifted around, right, by different mobile elements. And actually, mobile elements are a very important part of the genome of bacteria. And it might actually serve as mobile elements, meaning like plasmids or like phages that move DNA around. It might actually, I mean, this is like the most exciting thing to me that to think that these organisms, their genomes may be distributed amongst all these different related organisms,
Mm.
and there's these mechanisms that allow them to move around. And so here they do some sequence gazing at the genomes of this mycobacterium, which I think is important to study because it is one of these resistant, right,
Yeah.
antibiotic resistant bacteria. So like understanding like how it gets so antibiotic resistant is a way of potentially finding a therapeutic for it. I think that's one of the things that people might argue as the significance of such a paper. And yeah, and this is they find that they have all these mobile elements that move stuff around. Their genomes are the diversity of their genomes is actually you can track it to these mobile elements.
Yeah, there's lots of prophages and like various like segments. I think the at B like locate. And one things that I pulled out of this paper that got me thinking about bioengineering is because the amount of the amount of prophages and plasmids uh, can actually provide some tools for engineering this
Mm.
bacteria just in the lab to try and figure.
Oh, sure, research tools.
Yeah. So that's kind of where. But this is very much a bioinformatics genomics paper, but it does because I think it also boosts this idea of it's not just a genome, but like a metagenome of not just like you pointing out earlier the
Yeah.
I'm
All the
literally making the exact point you said earlier. But of
Yeah,
just
all those closely related bacteria are swapping genes. But it's also actually like in research, right? Like we're always trying to isolate certain things and then study them in their context. I think it also speaks to like when the tie to engineering potentially is just understanding that like these genomes, when you're trying to engineer like a bacteria that isn't like one that you made from the ground up, right? Like there's one engineering approach where it's like, oh, we're gonna build this thing where we know all the components and what they do. But
mhm.
if you want to take a bacteria in the environment and then bring it into a context where you're going to use it, right? Like you have to sort of understand that they have all these mechanisms for changing. So like if you make a change to them and you expect that change to stick over the long term, they may not want that actually, right?
Mhm.
Their genomes may not be set up to maintain a change in that fashion. And understanding the ways in which these mobile elements contribute to the changing diversity of a population is understanding those factors, right? What goes into making a change permanent.
Yeah, yeah, and I think I mean anybody who's ever had to like do genetic engineering on a bacteria, I know I'm speaking to a very narrow portion of our audience here,
Yeah.
who has actually had to suffer the loss of I've done this great mutation experiment, I've put my plasmid in, and next day nothing grows,
Yeah.
or you do a test and you find out wait it's not got the gene in the right way,
Mhm.
or like
Yeah.
the gene's in backwards so it's not transcribing that sort of thing.
Exactly. Yeah. Despite all your best efforts to get it in there and you see it's there, then you passage it a few times and you're like, it's gone, or something's happened to it that it's been inactivated, right? And it's because, yeah, I think in some ways we don't know, we don't have that fine-tuned knowledge yet, right, of
Mm.
exactly how the genomes work. And there's a little bit of a cut and paste mentality that we have that I think doesn't respect the total diversity of what's going on in those genomes.
Hmm. Okay, speaking of diversity, okay, this is a really tenuous segue, but we're talking about protists now. A free living protist that lacks canonical eukaryotic DNA replication and segregation systems. So, eukaryotes, they're like us, but these ones decide not to go the multicellular route. They decide, no, plants, boo, 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 humans, boo. Let's just be on our own for a bit. They're the loners of the eukaryotic world. Um,
They're perfectly fine just swimming around by themselves and not coming together in clumps or anything.
So yeah, what they did was this study they did a draft genome assembly for this free living metamonad called Carpe Diemonas membranifera.
That's a hilarious pun sort of name.
Yeah, Carpe Diem, seize the day.
Yeah.
Yeah, and they did some sequencing for this. What they found was so for a lot of metamonads like this, they're kind of parasitic. They found that some of the replication genes are missing,
Hmm.
and so the genes that usually govern mitosis, which is quite an important thing for our cells because they need to like get everything needs to be put in order for the chromosomes to line up and for all the kind of DNA to. And what they found was that they completely lacked things like the origin recognition complex and other like big reposome components. So
Hmm.
so this is almost like interesting. So almost reflecting what we did talk about last time about finding something a new bacterium and well
Yeah.
not a bacterium a new life form and seeing
Yeah.
that it's doing something completely different to what we're
Single
thinking of.
celled life form.
Yeah, single celled life form.
And I
Um,
think one of the analogies I didn't deploy at that last in that top 10 one, but I think it's reminding me of it as we're talking about this paper here, is that this is like the microbiology equivalent of like going out into the savannah and like following around a lion or something and being like,
yeah,
oh, we found out this lion does this and this and this and it likes to eat these things. Like this is very much the same sort of action. It's just that like you're looking very small and the features are all these molecular features, right? Like, oh, it seems to have these genes and it does this sort of thing with them.
yeah. And the interesting thing for this is not just is the kind of bioprospecting aspect, which is we found this lion and that pelt looks like it'd be really good to wear. So the bioprospecting aspect of looking at all these like what they call extreme streamlining of the DNA replication process. So
Hmm.
it's interesting to note like what can we learn about because something different's happening there, and if something different's happening, that could be a potential like interesting new thing that we could learn about how to replicate DNA.
Yeah, I love these types of stories. I feel like, yeah, even and animal research is full of those instances as well, right? It's like,
Mm,
oh, like look at the way that this animal does this thing, right? Look at the way that it like, repels water off of its pelt, the way that it
yeah,
jumps. Like you get these lessons, and those same lessons that you learn from those bigger things, you can learn that from bacteria too. It's just it takes like a lot of different tools to get there.
yeah. And I think we're going to go to our final paper, which is I find quite exciting actually.
Yeah, quite hilarious, I think. I stumbled upon this one while looking through recently published things. And here we have microbially enhanced vanadium mining and bioremediation under micro and Mars gravity on the International Space Station.
Yeah,
So
there's
I think this is once again embracing the idea that microbes are our tools, right? And that we
mhm.
use them for all these different applications. So in mining, they use microbes to do bioremediation and also mining itself. Leaching, bioleaching, I think is one of the
Mhm.
ways that they call it. And it's microbes that get spread upon the ore, and then through their metabolic activity, they kind of solubilize some useful metal ions, and if you let that go for long enough, you can create like these big pools that are enriched with the with the metal of your choice.
Yeah,
But does it work in space?
yeah. This is the cool part of it because space has lots of unique challenges doing research. So part of the interesting thing about this paper is seeing how they actually because they have like a various like interesting setups for how
Yeah,
so
well, I
like
think this is very common for space research because right, astronauts they're trained in many different things, but
mm,
like they're not like intense experts in the particular experimental design, right, that someone's trying to deploy. So they often build like a contraption where the experiment takes place, and then the astronauts become the operators of the contraption, right? And the data will then get like analyzed at a different point in time. And so they've
yeah.
built like yeah, they call it the BioRock experimental unit in this particular paper, which allows them to expose pieces of rock to different types of bacteria in the International Space Station where they can control gravity, right? Because that's a big thing that would be different if you're trying to deploy these these biomining solutions, right, in space or on a different planet, is that the gravity would be different, and we don't know whether or not all those assumptions that we have made and the way things work are dependent on that.
Yeah, so that's the. I mean, they test three different bacteria. There's, yeah, there's. It's really interesting because I mean, Palmy's like they also ran like concurrent experiments on the ground as well, which I think is really important as well. Just having a good control and looking at the effect of microgravity. Admittedly, at the end of it, they don't find much of an effect of microgravity, so you could argue that maybe they didn't need to put these bacteria up into space. But come on, why wouldn't you?
You don't know it until you try. Um,
Exactly.
but I mean, at the end, I mean, I guess this is interesting because I'm not really super familiar with this field of just like testing microgravity, right? That's the simplest like
Yeah.
experimental difference between running one experiment on the ground and one in space, right? Is you're testing like what is the difference of like having that gravity there. Maybe they could have predicted this, right, from like other microgravity experiments where it's like it doesn't seem to affect bacteria in this way and that, but actually it might not, right? I think that that's why the experiment was done. And of course you cannot hide this data away because it took so many resources to do, and it's international collaboration, right? If we're using the space station as our lab, so yeah, some interesting
Yeah.
data there in the
I mean,
world.
there was like a study a couple of years. I mean, this is so long ago. A study conducted on the space shuttle. It was that long ago, but on like bacteria changing, becoming slightly more pathogenic or growing slightly different in microgravity. So I think there is still an aspect of microgravity we maybe don't necessarily understand on the effects of bacteria. So I think it is worth trying exploring that.
Yeah,
Um,
I mean, from first principles in my mind, I could already say that like it was useful to do because like when you think about bacteria interacting on a surface, right? That's all about attachment and like how close those bacteria get, especially when you don't necessarily know the full mechanism of what drives that phenotype. So in space, does attachment work the same way? Is bacteria floating around going to do the same stuff? add to that, like, is their metabolism still going to be the same, like that it could do this type of reaction on the surface of the rock? So yeah, I mean, there's plenty of reasons. It's just like it does seem kind of excessive almost, because I don't know how close we are to actually mining vanadium in space.
I mean, theoretically, I don't think we're that close yet. But I think that our opinions on this would be completely different if they had found a difference. That's the thing. That's why I think this is worth pursuing.
So,
Like, the only
mhm.
reason we're writing this is because they didn't find anything. But if they did find anything, then they'd be doing essentially the same thing. But
Yes,
so I
yeah,
still think it
yeah,
is definitely worth it.
yeah. So yeah, those are our selections. What are we feeling for covering on our deep dive? Um,
Hmm.
I like, I like, I like the swarming bacteria and the single strain control out of this group. Um, I like the infection of the oral cavity for SARS-CoV-2 infection, just because
Hmm.
it is all that single cell sequencing is kind of fun to talk about. It's like, yeah, something big that's happening. I'm not sure what else
Yeah.
I feel. I actually don't really feel like talking about spatial landscape of lung pathology during COVID-19 progression.
Yeah, I'm kind of feeling the swarming paper
Mhm.
because I feel that made a very good impression. It looks like it'll be quite interesting. Um, I'm also quite interested in single strain control of microbial consortia. Um,
That one is more that one's definitely more model focused, right? There's a lot of
yeah,
mathematical stuff that they're doing. So maybe swarming bacteria is a little bit closer to the experimental like setups that we'd be more comfortable in. I'm a little bit mathed out from talking about all that deep learning last week.
oh, yeah. I mean, I I feel you, but I also have to warn you that there is biophysics in the eLife paper, in the swarm paper as well, where they talk about the biophysics of collective motion in bacteria.
Oh,
So
okay. So yeah, so
so
that's like yeah, like the physical properties of like what happens at like the edges versus the insides. Yeah, some
Yeah. So there
friction.
is a lot. So the idea is that almost like when the bacteria form these swarms, the antibiotic causes them to form into a traffic jam of some sort. And then that's what causes them to form a biofilm. So it's almost figuring out those properties that cause it to form in a traffic jam. Then, so yeah.
Things
The thing
those
is,
are linked to life, right? Like those are linked to like these are processes that exist in bacteria, right?
yeah,
There's something that like machine learning is not grounded in reality in the same way that I feel this is. That's speaking
yeah.
from a microbiologist standpoint.
I mean, you can already tell I'm biased in favor of this paper because I did some extra reading on it to get come up with that analogy.
Let's do it. Let's do swarming bacteria undergo localized dynamic phase transition to form stress-induced biofilms.
Yeah, okay, I think that's a good plan. I quite like that paper. That means we have lots of videos to show next week. That'll be quite good. Um, yeah,
uh, yeah, so join us next week for our deep dive week, where we cover swarming bacteria undergo localized dynamic phase transition to form stress-induced biofilms.
and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible that we didn't get everything right. So science is about thinking critically and about asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach us out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is this process where we can all participate, and we hope that you've all had a good time listening to us to ramble on about microbiology today. If you think you have something to add or found something unclear, just let us know.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content.